Hemos becado a más de 1 millón de estudiantes universitarios.
Más de 84,000 alumnos y alumnas han sido beneficiados con becas nutricionales.
Lanzamos una campaña para apoyar a la Universidad Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA.
Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo Val, más de 2,000 universitarias han sido beneficiarias.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un Kit y Apoya a un Residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la Reserva de la Biosfera de Jano, Chihuahua.
Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres.
Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Martes.
Apoyamos a alumnos de escasos recursos.
Asignado a la opción señalada, institución de asignación UNAM.
¡Sí quedé! ¡Sí quedé!
El sueño se cumplió. Ahora formas parte de la comunidad universitaria y puedes acceder a las becas que Fundación UNAM tiene para ti. Conoce nuestros programas en www.funam.mx y en nuestras redes sociales.
Bienvenidos de conferencia, dado por acompañen. El día de hoy tenemos por que nos acontece la invitan nuestro ético y esto una de las cosas más pues un campo siempre enfático se ha caracterizado porque en un periodo muy corto ha sido un muy amplio y las capas ahora permiten ver detalle muy poco, muy estable. En la conferencia es profesor en la universidad, en especial. Welcome the University and Seminar Series organized on Fundación, the Colegio Nacional and the Consortium of Universities for Science. The conference today is by Professor Vagheesh Narasimhan de la Universidad de Texas en Austin, del departamento from the Department of Integrative Biology. Thanks very much, Vagheesh, and glad to see you. And the conference and the cycle are on the human evolution through the studies on paleogenomics and genetics. This research field is characterized by a very rapid development. In a few years, we are now studying questions that just a few years were even unthinkable, were very difficult to even start doing this research. So we are extremely happy, Vagheesh, and we are joined by the president of UNAM Foundation, Dionisio Meade, the executive director of the UNAM Foundation, Araxi Rodriguez, and the president of the Universities for Science Consortium, Araxi Urrutia, and Mashal and Vagheesh. Thanks very much. All means nuevamente conferencia Texas RS.
Muchas gracias, Jaime. Muchas gracias a Mayo y a Araxi. Bueno, esta serie de pláticas sobre temas tan interesantes y en donde estamos viendo las capacidades de la paleogenómica, todas alrededor de esta ciencia, no diría yo que es reciente, y cuyos avances recientes son muy novedosos, muy ilustrativos, enseñan mucho de ciencia humana. En este caso, la evolución desde los ojos, las lentes, DNA. Muchas gracias a Vagheesh por su Marshall nuevamente. Subrayar que nosotros, Consorcio, se ha vuelto un instrumento fundamental. Nos permite contar con la cooperación de académicos, de investigadores prestigiados de muchas de las universidades que se han sumado a este consorcio y cuyos expertos, como en este caso, nos presentan sus avances, nos presentan sus logros, nos presentan lo que ha traído consigo la investigación alrededor de este campo. Eso significa para nosotros una gran motivación porque abre los ojos a muchos de nuestros jóvenes que están por definir su perfil profesional para que encuentren en esta parte de la ciencia un campo adecuado para poder realizar su vida profesional. Quiero mencionar que la Fundación es en México el único organismo que forma parte del Comité Asesor Nacional a las ciencias básicas. Y justamente lo hace por considerar que a través de presentaciones como estas estimulan la investigación y que para nosotros eso, como lo hemos comentado en ocasiones, es una actividad, bien es la forma que hemos comprometidos a la altura del conocimiento. Así que agradecemos mucho la presencia de todos. Estamos ya a nuestra mejor disposición de presentar este tema tan todo lo que la paleonómica siendo conocimiento. Muchas gracias a todos y pues le damos la palabra a Marshall para que nos presente a Vagheesh y luego a él para que nos haga su presentación. Muchas gracias a todos y pues aquí estamos a la orden.
Muchas gracias, Jaime y Dionisio. Entonces, hoy tenemos el placer de tener al doctor Vagheesh Narasimhan. Dr. Vagheesh se incorporó a la Universidad de Texas, Austin, como profesor de departamentos de Biología Integrativa, Estadística y Ciencias de Datos y Salud Poblacional en otros. Él obtuvo su doctorado en Matemática y Medicina en la Universidad de Cambridge, Wellcome Trust Sangre. Posteriormente realizó su investigación postdoctoral en el Departamento de Genética de la Facultad de Harvard con David Reich y Nick Patterson. Vagheesh se interesa por los que en la informática y la. Today we have the great pleasure Austin started as a professor in integrated biology science. Fall 2020, received his Cambridge and did his postdoc work at the university. Interested in problems of detection of. So last week we learned about paleogenomics, about how we paleogenomics understand human. La semana pasada aprendimos genómica y hoy podemos usar la paleogenómica para comprender. Muchas gracias.
Awesome. Great. First of all, muchas gracias. Thank you for having me present some of the work we've been doing in the laboratory. So I want to begin maybe with a little bit of recap of what was presented last week. I'm not even sure who presented that, but I thought it might be worth talking about a little bit about ancient DNA because it's all possible. And ancient DNA, of course, is a magical enterprise which allows us to extract DNA or genetic material from skeletal remains thousands, if not tens of thousands of years ago. And so the basic process involves the excavation of archaeological remains. Here is a drone image from a site from around 2500 BC, and from which bones are taken into the laboratory. And these were images taken from my postdoctoral laboratory where individual bones were brought, and portions of these bones, particularly the cochlea, are extracted from the bone itself and then are sequenced in specific protocols that allow this to be possible without contamination. And contamination is a major issue with ancient DNA because there's DNA floating all around us. There's DNA in our skin, DNA in our saliva, DNA in the air, and so on. And it's very important that the DNA that's floating around in the environment which is often 1,000 times in higher concentration than the actual DNA remaining in a sample, does not contaminate the overall sample. And so the extraction of these DNA samples occurs in very specific conditions in a clean room, which has specific separate airflow into the room. It has positive pressure to push out particulates away from the sample. The extraction is often performed in the presence of UV light. The surface where UV light destroys DNA from the surface. Sometimes samples are bleached on the surface if they've been handled to remove DNA from the surface of the sample, after which 75 milligrams of bone powder are sandblasted on the sample and then prepared into next generation sequencing libraries, which are then sequenced by the sequencing machine. So we're able to go from skeletal material thousands of years ago into genetic material that can then be analyzed. So there's been a huge explosion in the availability of ancient DNA data over the past 10 to 12 years. In the public domain, there's maybe, this is only since 2019, but in the past 5 years, maybe about 10,000 samples have come online. And so this has been a major explosion, almost 100-fold increase in the availability of ancient DNA data that's been available to analyze. So one of the most amazing things that has happened is the expansion of ancient DNA data, not just from one geographic region in the world, Europe, but now an expansion of ancient DNA data to many regions of the world. So in 2019, I led a study that was the main analyst of a study looking to expand the sequencing of ancient DNA, which had been largely confined to the geographic area of Europe, to expand to different parts of Asia, where we managed to get additional samples, and to then look at the overall genetic history of Eurasia, to then answer several questions around both demographic change, but also a change in the genomes of individuals living in this location. So over the next 30 or 40 minutes, I'm going to talk to you about this endeavor, which has been a 5-year or 6-year endeavor from my point of view, beginning first in 2019, when I was involved in the collection and sort of publication of a large number of these samples, and then utilizing these samples to understand genetic change over time. And understanding demographic change and understanding change over time go hand in hand. And so I'm going to talk first about inference of demographic change, and then talking about how we can use that to understand changes in natural selection in human populations over this time period in this geographic space. So in 2019, we published a paper involving the sequencing of 523 new individuals. It's one of the largest ancient DNA studies published to date in a single publication, where we reported a huge number of individuals from Asia, complementing a large number of individuals already sequenced from Europe. And our main goal was to ask 3 questions that sort of fall on top of each other. One is, how does modern genetic variation in Eurasia arise? That is, how is genetic variation distributed? Second, if we know how that genetic variation is distributed and know how that changes over time, how can we use that this information to understand natural selection? And third, since we have so many genomic samples currently available in the tens of thousands, can we use that to inform how samples might be sequenced into the future? That is, can we understand how ancient DNA preserves over this landscape and use that to answer questions about which samples we should sample from into the future? So I will be talking about all 3 things over the next 30 or 40 minutes. So I want to begin by talking about the population history of Eurasia over the past 10,000 years. And the population history of Eurasia is markedly changed really by a fundamental process that occurred in the past 10,000 years. That is the advent of farming. The advent of farming is thought to begin, at least in Eurasia, somewhere in the Fertile Crescent, at least wheat farming, and then begins to move into the peripheries of these continents over subsequent millennia. So here what you're seeing in these colors, in the darkest color, are where farming is known to exist from archaeobotanical evidence, that is grains that are found in the soil at different layers, where different layers represent how long ago those soil samples are from. And so if you look at these layers, you find archaeobotanical evidence for agricultural practices, first beginning in the Fertile Crescent around 10,000 years ago, and then it slowly begins to expand over the next several thousand years into the peripheries of Europe. So an open question in the field of paleogenomics and indeed understanding human history really has been whether the movement of people has, whether this change in farming technology and the advent of farming across this landscape has emerged due to the movement of technology. That is, do people from one part of the world realize that, oh, look, you can carry out this process called farming and they get these grains and seeds and start to grow them in different areas? Or is it driven by the movement of people? That is, do people from one region of the world move with their technology and then start to practice this particular cultural change, which is farming across these regions, and replace people who lived there before, who are largely surviving off hunting and gathering practices? So this was an open question even from about 5 to 10 years ago that has largely been addressed through the sequencing of ancient DNA, because you can now obtain DNA data from all these parts of the world across these time periods, and then look at the genetic change over this time period to try and understand whether it's the movement of people or the movement of technology that's driving this sort of cultural transfer. So in order to do this, we need to understand or develop I'm sorry, my video got cut off. I'm going to join again. Can everyone hear me again?
Yes.
Okay, sorry. I have no idea what happened. I think my internet just cut off. So in order to understand change over time and to understand the process of mixture, we needed to develop new methods to understand how populations mix with one another. So here's an example of four populations, 1, 2, 4, and 5, and population 3, which is a mixture of populations 2 and 4. And this is one example of mixture. Another way these populations could have mixed was you had populations 1, 2, and 4, where 4 is drawn from a lineage that comes from the side, from the left side, which mix together to form population 3. Another type of mixture model is one where you could have a mixture initially of population 4 and 5, and then you have a mixture with 2, leading to population 3. So we needed to build methods that can look at this complexity of mixture and model them and try to understand which populations are mixing with what others to give rise to new populations that lived later in time. So along with trying to understand which population mixed with one another, we also need to understand when in time this mixture happened. So here's an example of population C being formed from population A and B, and we want to understand the time in which this mixture happened. And so we've built methods to do this, and the main way in which we infer whether mixture has happened or not is to look at the segment length of DNA ancestry that's present in an individual. So here's an example of an individual who is an offspring of Neanderthals and modern humans. So in this case, the person here has one ancestry from a blue chromosome, that is from a Neanderthal ancestor, and one from a gray chromosome from a modern human ancestor. And then in the subsequent generation, through the process of recombination, this blue segment is broken up into a smaller section. And several generations after inbreeding, these sections are broken down into smaller and smaller windows, giving rise to the idea that the length of these segments provides information on how many generations ago mixing has happened to give rise to that particular individual. That is, we can infer not just whether mixtures happen between Neanderthals and modern humans, as we've built models for in this side, but we can also infer when in time mixture happened. A third thing that we needed to do is to be able to perform Y chromosome calling. That is to try and understand the paternal and maternal haplogroups or ancestries that are associated with individual samples. And as part of this process of generating these sequences and to try and understand population movement over time, we needed to infer whether mixture has happened, when in time mixture has happened, and whether this mixture occurred with maternal or paternal bias. And so I'm going to talk a little bit about the sort of population history of Eurasia using the methods we've built to sort of look at all these three questions. Over the next 5 minutes, I'm going to talk about the population history of Eurasia over the past 10,000 years. And this has largely been driven through the accumulation of ancient DNA data, for which we have ancient DNA, not just across space, but across time over the past 10,000 years. So around 10,000 years ago, populations in Eurasia were divided into different hunter-gatherer groups that broadly lived in different parts of Eurasia. And so these are indicated by these colored rectangles. And in these gray or beige polygons are the extent to which that ancestry is thought to permeate. And what I mean by this is that if you take individuals from a part of Europe where these beige polygons exist, these individuals share a common ancestor with each other before they share a common ancestor with individuals from these orange, red, purple, green, and teal regions. So that is hunter-gatherer groups that existed from around 8,000 BC or 10,000 years ago, were largely separated from each other for tens of thousands of years, and largely lived in these isolated groups where they shared ancestry within a small geographic region, but didn't really share ancestry across these sort of beige landscapes. So at around this time period, a major change occurs culturally, and that is the advent of farming in the Fertile Crescent around 10,000 years ago. And this is largely driven by the advent of wheat and barley domestication in this region. And what happens is we can then look to see how this ancestry that's seen in orange, blue, red, and purple, and so on, change over a time in which agriculture starts to move across this landscape. So if we move several thousands of years later, so if we move from 8,000 BC to 4,000 BC, what we see is a change in the ancestry inside these beige polygons that existed before. So if you look at Europe, you see a very clear change that's happened. So from 8,000 BC, where these circles, which represent ancestry that was present there, was largely blue, now we have orange ancestry, which previously only existed in the Fertile Crescent area, has now moved in, suggesting that a large proportion of the ancestry of people living in Europe had been replaced by the movement of farmers from Anatolia into Europe over the next 4,000 years or so. That is the advent of farming in Anatolia and further subsequent, the development of agriculture in Europe has led to major mixing between the hunter-gatherers who lived there in Europe previously and farming populations who moved in from Anatolia over the next several thousand years. And this is true not just, of course, in Europe, but also in other parts of the world, in Central Asia and so on. And at the same time as the development of agriculture and farming practices, there was also the development of pastoralism that occurred slightly further north. And as part of this process, again, there was a mixture between hunter-gatherer groups. And so in the north of Eurasia, there's a mixture of different pastoral groups and different hunter-gatherer groups that existed before. And in the south of Eurasia, a mixture between different farming and hunter-gathering populations that existed there before. So at around 3,000 years ago, that is after the advent of farming, there's already a huge mixture of different hunter-gatherer populations that existed across this landscape. So around 3,000 years ago, another major cultural change occurs in Eurasia, and particularly around this northern part of the Caspian Sea in the Pontic-Caspian steppe, where there is the advent of the inventions of wheels and wagons and also horse domestication. This allows movement of people at very rapid scale across the landscape of Eurasia. And over the next several thousand years, particularly in the Bronze Age, so by this time bronze is already invented, there's major movement of these individuals who domesticate horses and also invent wheels and wagons across the continent of Eurasia, further spreading additional ancestry. And this process, again, takes several thousands of years, first beginning in sort of the Eurasian steppe, and then migrating slowly over into the peripheries of these continents over the next 1,500 years or so. So the basic process of the past 10,000 year history of Eurasia really is this two-step process where hunter-gatherer ancestry in blue, it mixes with farming populations that arrive here in orange. And then in the Bronze Age, a third population, a pastoralist ancestry arrives, thereby leading to this tripartite ancestry of Europe and South Asia, where individuals have different components of these three different ancestry groups that are present across modern individuals who live in this region today. And individuals from different parts of South Asia and different parts of Europe vary according to these proportions, but by and large, all individuals from these regions have all three types of this ancestry. So one of the most interesting things is if we look in Europe, the type of ancestry that persists over time. So here's a paper from a colleague of mine, Olave et al., where he shows the proportion of ancestry from the steppe pastoralists who arrived in the Bronze Age into Britain at around 4,500 years ago. So in blue are individuals, or here is the proportion of steppe ancestry on the y-axis, the pastoralist ancestry. And so it changes very rapidly at this time period around 4,500 years ago, where the last stone goes up at Stonehenge. The last blue stone, that are these big menhirs, go up at Stonehenge. And so you have a major population transition in the Bronze Age, where about 90% of the ancestry of the people of the British Isles is replaced by the movement of these pastoralists coming in. Another very interesting change that happens is a change in the Y chromosomal ancestry. That is the paternal ancestry that's present in the individuals before and after this mixture. So if you look at these colors, they show the paternal ancestry of individuals before and after mixture. And so what you're seeing here is that there's a major change in the paternal ancestry of the individuals along with the overall ancestry in the British Isles. In Spain, there's an even more dynamic shift where only about 40% of the overall ancestry is changed by the movement of pastoralists, but there's about 100% change in the paternal ancestry of individuals across this landscape. And so we were able to look also not just at whether mixtures happened and when it happened, but also the type of mixture that has occurred, and showing that it's largely male biased, at least in Europe. Another very interesting thing that we're able to see, thing we're able to see is inbreeding levels over time. That is, and this is a measure of population size occur in Europe over this window. And so what you're seeing is that the population sizes in Europe are greatly expanding over this window, leading to the present day. And we infer this by looking at regions of the genome that are so-called identical by descent. That is, if you have two parts of your genome that are exactly the same as each other, and that is you get the same copy from your mother and your father, we call that a homozygote. And so regions of the genome are inherited from the same ancestor or homozygotes and have some measure of what's called inbreeding. That is, if you have individuals whose parents are related to one another. And so if you look at this, this shows that over time, in Europe, people's parents are less and less related to one another, thereby suggesting increases in population size over time. And we can also observe this using another measure called FST, which looks at population differentiation. And so what you're seeing here is that in the Mesolithic, that is the Stone Age, populations were highly differentiated compared to each other. And then over time, they become less differentiated because of mixing that's happening across this time period between groups that are farming and hunter-gathering and so on. So the genetic information that we're seeing from these admixture profiles, it's also being reflected at the level of individual genomes over this time period. So another major thing that we can observe is dietary change through isotopic information that's available from these individuals. So you can extract not just DNA from these individuals, but you can take a tooth and then look at its carbon and nitrogen isotopes in that tooth. So one of the amazing things that's possible from isotopic information is to try and infer some form of dietary change that's occurring. So nitrogen isotopes in particular track the trophic level of an organism, and trophic levels basically mean where along the food chain you're getting your nutrition from. So if you're a carnivore, that is, you're only eating animals that are then eating other animals or indeed eating plants, then you would have a very high trophic level and high nitrogen levels. But then if you're largely eating plant-based foods, you expect to see lower nitrogen levels. And what you're seeing here is from the hunting and gathering period to the farming period, you have a major transition in diet where you change from largely, where you're eating a highly meat-based diet to one that's more starch-based. And then after that, you have slowly ramping up of additional meat-based diets as we get to the present day. So there's a major change that goes on in culture between the hunting and gathering period and the farming period. So I told you a little bit about what we understand about the population history of Eurasia. That is, it comes about this tripartite structure of mixture between different populations. So how can we use this to understand genetic change over time? So one of the ways we can do this is to use the mixture process itself. So here's an example of how we do that. So we have a population C that arises from population A and B, with 20% mixture from population A and 80% mixture from population B, giving rise to population C. And so what you can see here is you can take a single mutation, and in that single mutation, the frequency of that mutation in a population A is around 50%, and in population B, it's around 10%. And since population C is a mixture of population A and B, the expected value is that you have a weighted average of the allele frequencies, 50% times 20%, and 10% times 80%, which is around 18%. And so the allele frequency of the daughter population here, C, should be a mixture of the mixing fractions of the individual ancestral populations. However, if you observe several generations later that the allele frequency is drastically changed, then that's an indication that natural selection has occurred after the mixing process has happened. So we can use the mixture that has happened over the past 10,000 years itself that I talked about to understand whether selection has happened based on the allele frequencies of the parental populations that gave rise to that mixing population. So when we can do this concretely in Europe, where we have three different types of mixtures that are happening. First, the mixture between hunter-gatherers and farmers that move in. Second, from pastoralist groups mixing with the farming populations. And third, a homogenization of the farming and hunter-gatherer populations and the pastoralist populations in Europe, largely in the past few thousand years before present. So we're breaking down the sort of population history of Europe into three segments. The first is the Neolithic, that is the transition between hunting and gathering and farming. The second is the Bronze and Iron Age, between 6,000 years to 3,500 years. And finally, the most recent 3,500 years. So I want to illustrate this example with looking at a marquee, or illustrate this looking at a marquee example of one type of an event of natural selection that's happened in humans that's widely thought to be the strongest signal of natural selection in modern humans. That is lactase persistence, i.e. the ability to digest milk into adulthood. So if you look at this map, this is the population prevalence of this particular trait, the ability to digest milk into adulthood around different parts of the world. So if you look in Northern Europe, it's almost around 100%. That is, everyone from this part of the world is able to digest Europe into adulthood. but then it follows a gradient, which largely follows the proportion of steppe ancestry or pastoralist ancestry that's present here. So one question is, when is natural selection for this occurring? And we can use ancient DNA to directly address this. So here is the allele frequency of this particular mutation that gives rise to lactase persistence in these populations, both in Europe and in Central and South Asia. And what we can do is divide population history into these three time windows. One is the hunting and gathering farming transition, the other is the Bronze Age, and finally the historical period. And what you see here is the major change occurring only after the Bronze Age. And indeed, the allele largely rises very rapidly only in the historical period. So we're able to divide the population history of Europe into these time windows. And by having allele frequency information here, we can directly infer when selection is happening across this time window, like I described before, using the mixture process itself to infer natural selection. So this is an example just of lactase persistence changing over time. But one thing that's interesting is what other alleles are under selection over the past 10,000 years. So this is the change that occurs between the hunting and gathering period to the farming period. And what you see here are our 22 chromosomes on this map. And that's the x-axis. And each of these positions here represent individual mutations in the genome that are changing in response to natural selection occurring between the change between hunting and gathering to farming. And on the y-axis is the strength of our ability to detect selection in each of these loci. So I'm going to talk not about every single locus that we've discovered today, but a few marquee loci that we've that I want to talk about and discuss and provide some evidence as to why they might have been under selection. So the first locus I want to talk about is this gene FAM49B. It's a gene that's thought to, or known actually to confer resistance to salmonella infection. So a paper from 2019 in Nature Microbiology showed that mice lacking this particular allele or this particular gene were much more prevalent, or much more susceptible to salmonella infection compared to mice who had a functioning copy of this allele. So this is a locus. This is called a ++. That is, these mice contain a working copy of this gene, and then these mice do not contain a working copy of this gene. And they were able to use bioluminescence photography to look at regions in the genome, or regions in the mice body that sort of sequestered salmonella. And so here you see that when they were infected with salmonella, mice that lacked this particular gene were unable to sort of combat the salmonella infection presented with it, but individuals who had this allele did not. And in this, on this plot, there's an infection challenge assay where they infect these mice at day 0 and then observe in the spleen and the liver the presence and the extent of salmonella replication that's present in the spleen and liver. And so individuals lacking this gene have a much higher presence of replication of salmonella inside their bodies. So that it's suggesting that FAM49B has a causal role to play in combating salmonella infection and preventing replication of salmonella in vivo in model organisms. At the same time, as we found evidence for natural selection at this locus, a group published ancient DNA evidence for salmonella ancient DNA. That is, they sequenced ancient DNA not just from modern humans, but also from bacteria. And here they were able to sequence salmonella DNA strains from a range of different hosts. Some of these hosts are domesticated animals. And what they were able to do is to look at what happens at the hunting and gathering to farming transition. So they were able to look at individuals who largely were foragers, that is, they were hunter-gatherers, or individuals who were not practicing farming, and individuals who were practicing farming here in yellow. And what you're seeing here is that individuals who were practicing farming here in yellow were infected with the lineage of salmonella that came from domesticated animals, here pig, But individuals who are practicing foraging were obtaining salmonella infection in their bodies from other sources, including porpoises and horses. So that is the transition from hunting and gathering to farming results in an acquisition of salmonella strains into the genomes of these individuals at the perfect time when they're beginning to domesticate these animals and live with these animals and indeed with closer to each other. Another thing that this paper observed in these red lines are what they call salmonella infectivity islands. That is regions of the genome in the salmonella genome that allow salmonella to become specifically infective to humans. And so the acquisition of these elements of bacteria that allow them to infect specifically humans occurs specifically during the hunting and gathering to farming change. So what we're seeing in composite is a gene that essentially allows humans to be able to combat salmonella infection and clear this infection more quickly than previously. And during the hunting and gathering to farming transition is when humans are first infected with salmonella from domesticated animals, and the salmonella itself undergoes change in its genome, allowing it to infect humans. So we're observing at least a signal of host bacteria co-evolution, where the bacteria is evolving to become infectious to humans, and humans are evolving to become more resistant to this particular bacteria. So the hunting and gathering to farming transition is driven largely by mutations that allow us to sort of resist different bacterial infections and viral infections that are occurring during this major shift. Another change that occurs is in this gene FUT2, where experiments have been conducted in 2005 and later, where they show individuals who are infected with norovirus infection, and where individuals with the capital S have this allele, and individuals with the bottom S, individuals with the bottom S carry this allele in FUT2, and individuals with the capital S do not. And what you're seeing here is a contrast between symptomatic and asymptomatic carriers of norovirus infection in humans. And when you carry 2 copies of this FUT2 allele, it results in no individuals presenting with symptomatic norovirus infection, suggesting that individuals who carry this FUT2 allele secrete a particular secretory status in the blood that actually prevents them from getting symptomatic norovirus infection. So the transition from hunting and gathering to farming is also precipitates changes in the bacterial pathogen load that infects us and the viral pathogen load that infects us, and our own genome has shifted to this new environmental dynamic. So so that's what happens in the hunting and gathering period, and then we can step forward to the next 3,000 years and observe what changes are occurring in the Bronze Age. And what we're seeing here is actually mutations that are giving rise to slightly different types of change. So what we're seeing here is alleles that are largely associated with skin and eye pigmentation. So SLC45A2 and OCA2 and HERC2 are alleles that are associated with skin and eye pigmentation respectively. And what you're observing in the ancient DNA record here is if you look at the allele frequency of, say, this allele, which give rise to skin pigmentation over time, is that light skin pigmentation in Europe really begins to emerge only in the Bronze Age and then reaches fixation. That is, most individuals in Europe today have light skin pigmentation in the past several thousands of years. And in black is the expected allele frequency based on the mixture proportions. And so what you're seeing is that the allele frequency today is much different from the expected proportions that existed before. And so here is an individual, Cheddar Man. He's an ancient individual from around 12,000 years ago from Britain. And so at this time period in Britain, the individuals did not carry any light pigmentation alleles. So the sort of bust or recreation of Cheddar Man is shown here, where this individual has dark skin pigmentation alleles, which is very interesting because in Europe today, very few individuals of European ancestry have dark skin pigmentation. So this change actually is occurring very rapidly and only in the past 7,000 to 8,000 years. So in the third phase, that is in the most recent the 3,000 years, we observe, again, different signals of natural selection. And a large, and the major locus that occurs during this time period is lactase persistence, here LCT. But along with lactase persistence, we also identified many loci that are associated with vitamin D metabolism, suggesting that perhaps changes in diet were occurring across this time period and we needed different sources of vitamin D. And so there were changes that are associated with several alleles that are associated with vitamin D metabolism through this time period. One of the most interesting observations that we observed really wasn't in individual alleles, but on the overall type of alleles that were selected over this past 10,000 years. About a half of the total candidates we identified across this 10,000 year period were really found in immune-related genes and expressed in a variety of immune-related cells. So that is that the major, or about half of the selective pressures that affected us over the past 10,000 years were largely immune-related. That is related to the advent of bacteria, new bacteria and viruses that infected our genomes, and therefore our human body or human genome had to adapt to these changes in new viruses and pathogens that infected us. And we can see signals of this mixture that is present in our genomes. We also looked at natural selection, not just by looking at this mixture type process, but by looking at another process called haplotype homozygosity. So how this works is if you take a singular allele that's under selection, it often doesn't rise in frequency very rapidly by itself. It pulls mutations along with it up in frequency as it rises. And the way it does this really is because recombination, the process which breaks down genetic material, is not fast enough to break this down just to a single locus. And so individuals who are selected at this locus also carry with them regions of the genome around this locus, and there's a lack of genetic diversity around this locus. And so this provides a signature that we can capture to identify natural selection. So along with looking at mixture, we were also able to look at these regions which had lacked genetic diversity, and we were able to identify natural selection using an alternate selective scan. And what we found, actually, whether we used a natural selection scan using mixture or whether we use this alternate natural selection scan, we actually found a common message. And the common message is the following. That is, loci in the genome that are under selection at one particular time period, for example, here in the Neolithic shown in orange, are often seen in other time periods, but then other loci that are seen at other time periods are not seen before them. So there are some loci that are very specific to individual time periods, and loci that are really seen in the Neolithic often disappear as you go and you try to look to see whether those regions are under selection at other time periods. So that is, the process of mixture itself obscures signals of selection that had happened more distantly in the past, suggesting that ancient DNA is a powerful mechanism to uncover loci that had been under selection in the much more historical past that are no longer visible from genetic data today. So that's another message we're able to obtain from looking at these mixture processes and understanding natural selection. So finally, I want to talk about some new recent work we've done in the lab to understand how we might use all of the samples that have been sequenced to inform future sampling. And this is largely important, or very important, because of how destructive the process of ancient DNA is. So in order to get genetic material from these samples, we need to destroy the bones of these individuals that existed in the past. And these are precious remains. And we want to be as cautious and as careful as possible in sort of performing our sampling. And so if there's a way to determine before a sample is drilled, whether that sample would likely yield ancient DNA or not, and then preferentially sample or destroy samples that are more likely to yield ancient DNA, that could reduce the destructive nature of sampling. Another thing that we wanted to understand is what causes ancient DNA to preserve over this time period. Why do certain regions of the world, or why do certain time periods of the world, or what temperature conditions and soil conditions and so on allow us to obtain ancient DNA better than other regions and other time periods? And so is there a way we can utilize all the data that's been sequenced to try and get at this question? So we looked at this by looking at a singular data set of around 2,500 samples that were sequenced in 2 laboratories at Harvard Medical School and the Max Planck Institute of Evolutionary Anthropology and the University of Vienna. And what we did was to say that we binarized our data. That is to say, we sequenced 2,500 bones. 70% of these were successful. That is, they yielded authentic ancient DNA data. And we thought that we were able to obtain authentic ancient DNA data if it had 3,000 positions that we were able to capture on a genome-wide array. And what we looked at was what type of conditions might cause a sample to be successful versus not successful, and whether we can look at latitude and longitude and temperature, rainfall, elevation, and soil conditions, and so on, to kind of determine what determines ancient DNA preservation versus not. And so here are some curves that we can look at. And so what we can observe is that on average, annual mean temperature, the warmer the mean temperature is in a particular location, the poorer the DNA preservation. And here's the average SNP count, that is the amount of DNA we're able to get out of 1 million positions on our DNA capture array. And so what you're seeing here is that the warmer a climate, the less DNA we're able to extract. The higher the carbon percentage in the soil, the more DNA we're able to extract. The wetter the soil, the wetter conditions in a landscape, the less DNA we're able to extract. And of course, the older a sample is, there's less and less DNA left. So these are sort of gross scale measures of being able to look at sort of DNA preservation across these various climatological and other factors and soil-related factors that we were able to access. So another very important type of information that we had actually comes from imaging. So a study was carried out actually in 2017 to look to see if the preservation of the bones themselves contained information about whether a sample could be successful or not. And so they looked at around 34 samples. Some of them were considered poorly preserved. That is, the dental enamel and other types of bone material were certainly worn off, and others had really well-preserved root structure and so on. And they wanted to see if sort of the presentation was associated with overall outcome. Unfortunately, they only had 34 samples. So it was difficult to really power this study to look at whether image-based prediction would be useful. So we were able to look at this actually using novel methods in artificial intelligence, where we were able to look at bone material prior to sampling. And so we had bone material actually photographed in all of these laboratories where these bones were placed on an aluminum foil and then measured. And so what we were able to do is first crop the bone and just take the bone alone. And this is important because often some of these bags actually had information written down that, oh, this sample is from England, or this sample is from India, and so on. And so we wanted to remove the sort of labels and just use the imaging information from the bone directly. And so we segmented out just the bone. And what we did was to train an image-based convolutional neural network to sort of predict whether a bone would be successful or not. And we fused this neural network with an additional network, which was trained on metadata associated with bioclimatic variables, not just precipitation, age, and elevation, but soil, pH, elevation, and rainfall, and all these other sort of variables I discussed. So we were able to train a fused multimodal model, which took into account both image-based information and climatic and soil-based information, and pull this together into a single model to predict whether a sample would be successful or not. We were able to look at class activation mapping, which provides some interpretive information on where the part of the bone is actually informative. And this is not a very good image, but when we were able to look at this, we were able to identify the very spongy or porous parts of the bone, often presented as being highly predictive of whether that bone would be predictive of whether it would yield authentic ancient DNA. So we believe that we have an actual method to combine both of these types of information to yield outcome. So here's our predictive model. And so what we're seeing here is a raw curve, or area under the curve, which shows how successful we are at predicting whether a sample will yield ancient DNA or not. ResNet 34 is an image-only model. So if you look at the image-only model, you have a certain level of prediction. If you have a tabular model just using the climatic variables and soil variables alone, you have slightly better prediction, but combining the two yields the best possible result, suggesting that both climatic information and soil information are both informative at prediction. Finally, we were able to compare our models to an anthropologist who's able to visually look at this and also had information from the climatic information and so on. And so they were only able to predict whether a sample was successful or not with maybe 65, 66%. But we were able to do this with much higher accuracies and precision compared to previously because of the large data set size that we had and the ability to combine imaging and sort of climatic information. So I want to leave you with the fact that we were able to look at these three tripartite goals over the past 5 years. One is the population history of Eurasia over the past 10,000 years. Two is how natural selection, using that population history to leverage that to understand natural selection over this time period. And third, if you have this accumulated knowledge of all of these samples and whether samples worked or not, whether we can inform future sampling and help the field of paleogenomics by sampling samples that were more likely would yield ancient DNA later, rather than destroy these samples a priori. So with that, I'd like to stop and thank everyone for the ability to present, and most importantly, thank members of my group who sort of led a lot of this work and really made all of this possible. So thanks to my group and also for the ability to present.
Talk. So I'll start us off by, so one of the main things that you spoke about was how from learned that Europeans, but really have been a mixture of individuals that we refer to as hunter-gatherer, farmer, pastoralist. Can you comment on how that makes us think about Europeans or South Asians or Eurasians or people in general, and sort of generally thinking about human diversity when we recognize those layers of mixing that have happened over time?
Right. So I think, that's a great question. So one of the most, interesting and powerful observations that comes from paleogenomics is that we're all mixtures. So, even in very deep time, not just, looking at the past 10,000 years, but even in very deep time, individuals from Eurasia are mixtures between Neanderthals who lived in this landscape before and individuals from Africa who moved out from Africa into this landscape. And so this process of mixture is actually something that's ubiquitous and happened time and time again across the landscape of the world, really. And so Eurasia is no different. And so whether it's South Asia, whether it's Europe or Africa, whatever region, or the Americas, whatever region of the world we're talking about, there's been layers and layers of mixture over the past millennia. And it's only through the lens of paleogenomics that we're able to really examine this complexity and really understand what's happening. And so it's been a powerful way to understand mixture and also understand that mixture is a very ubiquitous, normal, and a process that happens constantly.
So in that sense, it's really, I think many of us, especially those who are maybe new to genetics, especially, may think that migrating and this kind of globalization is a modern phenomena.
Mm-hmm.
And so I think what you're showing is that it's actually been a part of human history for any time scale that we may look at, right? Which can be sort of a new piece of information for a lot of people.
Mm-hmm. Right. I think, I think it's even like talking about, 50,000 years ago, 100,000 years ago, Neanderthals, that's like something very far away. But, often when you're talking even about the past 10,000 years, people didn't appreciate this idea that even in very recent times, there's been so much mixture happening, for example, in the advent of farming, for example, in Europe. So that's a very cool new sort of discovery, I feel, that's happened. And we really can only do this through the lens of ADNA.
So in that sense, when we think today, or even when we say something like European, or I have ancestry from Europe, what do these results sort of mean for statements like that?
Right, so they're only referring to things that, are events that are happening maybe in the past few hundred years or something like this. So when people say, oh, I have European ancestry or African ancestry, they don't really mean ancestry that's present in humans from 100,000 years ago or like 200,000 years ago. They really only mean things that are very recent, like in the past 150 years in historical times. And, so when we refer to European ancestry or Native American ancestry or things like that, it's very, very recent. And we're not really talking about the types of events that I even talked about today, which refers to hunter-gatherers from Europe and farming populations from Europe, or farming population of Anatolia 10,000 years ago. So that's not what we're talking about. That's much more recent.
Great, thanks, Vagheesh. So another thing that you talked about was that about the evolution of lactase persistence, and you showed a map of the world with respect to that. So the evolution of lactase persistence is something that has been taken up in arguments by white nationalists and so on, but I think you gave sort of a broader picture. So can you maybe show that again and like comment on sort of how that's actually spread out throughout the world, and if it may be specific to populations, or if that's just a misunderstanding and it's really been in sort of soft sweep sort of in different parts of the world?
Yeah, so that's a great question, and I can show this here. So here is the prevalence of lactase persistence. So if you look at this, in red are world where the phenotype, the ability to digest milk, is present. And what is known now actually is that the locus that applies to each actually is different from each. So I showed that the locus that's actually common between Europe and, but in the Near East and in Africa, there are actually other mutations that independently arose at different positions of the gene that confer the same outcome. And so lactase persistence is a very interesting story and dynamic because they occurred at different time periods, parts of the world, through different mutations, all occurring independently, giving rise. So it's a sort of, in this work, I talked about what's happening largely in Europe. But of course, this type of selection is happening independently in the world, including in Africa. So, and they're completely independent and occur at different times, different dairying, and indeed different mutations is the ultimate source of giving rise to it.
So in that sense, would you say it's a complete misunderstanding that lactase underwent selection in Europe? And so why were studies sort of really highlighting that, like that sort of has flown?
I think it's just the availability of data. A large part of it was the initial studies came out from Europe, and so people thought that this allele was the same. It was the same allele that was present everywhere. But I think as data sets expanded and we realized that there were other locations in the world where this lactase persistence was present, there's additional understanding that the loci that's present in these regions is actually completely different. And so there's just a better and more complex understanding. And in fact, there's now a recent paper showing that this gene is actually under selection, not at the location that confers lactase persistence, but in another location in the genome in East Asia, beginning around 25,000 years ago. And this is associated not with lactase persistence, but it's thought to relate with infection and immunity. So that is that even in Europe, we believe that this could be due to dairying, but indeed it could also be due to other reasons, and that other reason could be the same as what's happening in East Asia, which is that this has to do with immunity. It's actually, we can infer whether regions are under selection, but it's much more difficult to make a case for why things were under selection. And we can do this in certain settings, maybe, within the case of FAM49B, where we have model organism evidence, and we also have evidence of infectivity islands in the bacteria, and we have evidence from the host. But in the absence of like this sort of tripartite evidence where we have a sort of piece of everything fit together, it's sort of difficult to understand why things were under selection. And so I think lactase persistence, we still have a little to think about. But I think, a large part of why the initial stories were painted in a particular direction is just lack.
That map, the places that do show very low level, why would that then, since a lot of the audience in Mexico comment also on about lactose.
Right. So, I'm not an expert on this, but I believe that lactose persistence tracks with the proportion of European ancestry. Actually, don't know much about it. So I don't want to overspeak or say something.
Fine, but in this map, though, areas like in Eurasia that are showing lesser, so you would say that's related to just less dairy consumption there in historical periods?
So, it's actually not clear because parts of Mongolia, there's been a lot of dairy consumption. And indeed, if you look at the proteomic evidence, even in Europe, dairy consumption begins much earlier than the selection of that locus. So dairy consumption actually begins around here. But it's only much later that the allele begins to rise in frequency. So it's not clear why, whether, maybe it's the processing is different. People are using dairy, but they're consuming yogurt or cheese, which is slightly different in its lactose levels. And maybe later on they're milk. So it's actually unclear what's driving that sort of selective, whether it's directly associated with milk drinking pressure. So, one interesting thing is, why does milk drinking have pressure? You can eat many different things and it doesn't have. And so people have postulated that perhaps during times of famine, this is the only type of nutrition that was available, and so therefore it rose in frequency. But I think there needs.
So another thing that you talked about was about skin pigmentation, which is also another really interesting trait with a lot of public interest. So you talked about it and showed how Europeans or people who lived in Europe tended to have darker skin. So if you could comment a bit more on what exactly is the evidence for that and what kind of factors may have led to selection for lighter skin, as you found in your studies.
Sure, that's a great question. So it's very interesting. So skin pigmentation is a trait that's slightly different from many other traits like height, in the sense that height is largely a trait that's driven by thousands, or variation in height is driven by thousands and thousands of mutations. Whereas with skin pigmentation, it's also driven by many hundreds of thousands of mutations, but a few mutations have a very large effect on skin pigmentation. So that is mutations on a couple of genes, SLC45A2 and SLC22A5, have major impact on skin pigmentation. That is, they really increase skin pigmentation to a major extent. And so this particular trait is more binary at some level than some of the other traits like height. And so because it's more binary, we can observe the change in the allele frequency of these mutations over time. And so if you looked in Europe over the past 10,000 years, you can directly observe when these two mutations really rose in frequency and became quite prevalent. And if you look at them, they only emerge after the arrival of Anatolian farmers from the Near East into Europe. And that process only begins in the hunting and gathering to farming period. And so the mutations themselves, which have these major effects on skin pigmentation, only begin to arise when the farming populations from the Near East move in and their selection for that locus. So the point one here is that European hunter-gatherer populations who lived in Europe around 10,000 years ago and before did not have this allele at all, and therefore likely had darker or dark skin pigmentation alleles. And this is what's represented in that bust of Cheddarman that's shown in the Natural History Museum. And a reason for why skin pigmentation could have arisen is vitamin D. That's one thought. So previously, people were living and practicing a particular diet, which is hunting and gathering, which might have, accumulation of dietary sources and nutrition from a variety of different spaces. But then they suddenly transitioned to simply farming. And so maybe that sudden change in just using a starch-based diet alone was not enough to absorb vitamin D for some reason. And so there was major selection for light skin pigmentation, which greatly boosts your ability to gain vitamin D from sunlight. And so that's a hypothesis. Of course, we can't directly prove it, but that's a hypothesis that individuals, and this tracks with the sort of latitudinal gradient. So even within Europe, individuals from Northern Europe often have lighter skin pigmentation than Southern Europe. And so the one thought is that vitamin D exposure might, or vitamin D uptake or metabolism might have given rise to this particular trait.
Great, thank you so much, Vagheesh. So what's next, sort of what's on the horizon in the next 10 years for ancient DNA research for Europe or for the world? Sort of what can we expect to see?
Yeah, so it's a great question. So I think one of the things that's happening really is the expansion of sample sizes, going not just, ancient DNA was a realm where we had singular individuals from one particular time period from one place. So we'd take a sample from 10,000 years ago and sample one individual there. And then we'd go thousands of miles away and sample another individual from like another several thousands of years later and so on. And so, but now ancient DNA has changed to a field where we're able to obtain hundreds of individuals from particular sites. So from one site, we're able to obtain hundreds of individuals. And this allows a different lens of understanding human history. So instead of looking at broad scale patterns of, oh, these are population movements at broad scale that are happening, we're able to look at fine scale movement, including fine scale movement happening within particular sites. And we're able to use information when we have lots of information from one particular site. We're able to gain two types of information. One is information about how individuals are related to one another. And in addition, relate these individuals to archaeology and anthropological evidence from cultural remains on the ground, and to understand social structure and other types of population dynamics that are present in the ancient past. And also to infer population sizes that were present from one part of the world at one part of time. So another thing that's happening, of course, is the expansion not just of samples from Europe, but also from other parts of the world. And so it's now going to be possible to study natural selection, not just from the environment of the past 10,000 years of Europe, but also the past 10,000 years of other parts of the world, where people were subject, of course, to different environmental pressures, different pathogens, different viruses, and so on. So I think it's a very exciting road ahead, and we're sort of investigating all of these areas.
Great, that's really exciting. So as you've also seen in your own work, and as ancient DNA has been moving more global, sort of what kind of ethical issues are there, if any, and sort of how have you sort of dealt with that in your own work as we're getting samples, for example, from Asia, different parts of Asia to start, and later on we'll be talking about Americas and India with more expert speakers, so they can also further talk about that. But in your own experience, how do you deal with that, and how's that?
That's a great question. Of course, it's very important. These are precious remains and sort of the cultural heritage of different communities, and indeed all of us. So we need to be very careful in how we're obtaining and sequencing and handling and reporting back our results, and also taking care of the remains and returning them back to the custodians of these sources. And largely we work with local communities, with museums, archaeologists and anthropologists who've been on the field and working with various communities and different cultural entities over this time period. My interactions largely have been in India, where there is a national service, the Archaeological Survey of India, which has a sort of centralized authority over the control and sort of dissemination of these samples, and also maintaining them and sort of curating them. And so they work very closely with archaeologists in different parts of the country during the excavations. And so we work in their sampling directly at the onset of our study, so describe our process and what we're doing very early, and sort of, as part of this process, also train individuals from these regions and work in partnership with them to develop the science. And so that's also a very important part of our process. not just carrying out the science, but also being able to work in partnership and train with individuals there and publish and do research with local communities on the ground. So I think the answer is all of it. Like, having a cautious, careful approach that's open and follows the most updated and recent ethical and sort of scientific guidelines is how we've proceeded and will continue to proceed.
Thanks, Vagheesh. So this is obviously something that scientists are really having to become aware of and work with, working with ethicists, working with archaeologists, anthropologists. So there's a lot of new space for transdisciplinary and interdisciplinary collaboration, and also like all the windows that opens and being able to do that respectfully, both in local and global context. So from what you're saying in your own work, there has been possibilities and ways of also training scholars locally in areas from which you're working from remains of. That's been part of the projects as well.
Mm-hmm. So we work, both not just with working with individuals, but also, actually having them involved in the present papers and presentations and the act of doing science themselves. And so learning how to build sort of admixture graphs and work with sort of the methodological things that we've done and sort of training and mentoring students to be able to do that so that they can then work with their own data sets and their own sort of excavations and carry their research out themselves going forward. So that's been a process that I've participated with, and fortunate to have been able to participate with.
Okay, so that's great. So now that we have Zoom and we have servers, basically it becomes very easy actually to train people across countries, across the world, and be able to use resources and share resources and data sets that way as well. So it's really a very exciting time to make science more global, more open, and to sort of break some of the barriers that have been present for a long time. So thank you so much, Vagheesh. Muchas gracias, Vagheesh. It's been a really interesting talk. Una plática muy interesante en que estamos ya llegando a ver qué podemos descubrir usando la paleogenómica, y en la siguiente semana ya vamos a escuchar de la doctora María Vela, que nos va a platicar sobre paleogenómica en el contexto de México en particular, que va a estar de mucho interés la siguiente semana. Muchas gracias, Jaime, Araxi, por participación. A pasar la palabra.
Thanks very much. Thanks, Vagheesh. A really very interesting presentation, and congratulations for your studies. And it's interesting to see how these human migrations have shaped our DNA code. Is really interesting. And I also found very interesting that well, there are also the environmental factors, the climate, even where they were living, and the conditions. So it's very interesting because it offers the possibility of integrating more detail.
That's totally right. Maybe one of the things that we actually began this idea of trying to apply is that in certain parts of the world, even though they had the same temperature, the same climate, temples would routinely fail, and that is fail to build ancient. And in other parts of the world, they would be quite successful. And so one question is why? And so maybe there's additional information. We've seen so many samples now that maybe we could try to teach what actually is oil or what's bone type and so on that might give rise to it or not, and will improve the sort of practice. That's our goal. So that is identify factors, maybe pH content or cation content, organic composition, like that. We can use that information to try and tweak the way in which we see samples, and thereby help to figure out whether there's something different in certain parts of the world working and extract DNA. And that's our goal. And if we can do that, that'd be very exciting. And even if we can't do that, I think in parts of the world where we do, yes, we can look at the morphology of the bone and to see whether that bone might actually yield ancient DNA and sort of preserve bones that likely would not, and only work on samples that might. So I think that's also a kind of interesting direction that we've been able to go and try to help the process of ancient DNA along with just using it to answer scientific questions.
Wow, very interesting and promising. Yeah, it's something it will be very valuable to do. Well, thanks very much, Vagheesh, and thanks, Mashal, and well, thanks to all who have been sending questions and regards. And we just reiterate the invitation Machal already did. Join us for the coming seminar. Thanks very much. Thanks, Vagheesh, and I hope to see you soon.
Muchas gracias, Vagheesh. Muchas gracias, Jaime. Es muy interesante, muy impresionante. Todo lo que se puede aprender del DNA. La secuencia, como ya Jaime, es bastante reciente. Que podemos irnos muy contentos de ese paso que haya dado, todo lo que puede traer a la evolución. Y ese es justamente el propósito de plática como la que tú nos has de compartir, que muchos de nuestros jóvenes encuentren en estos espacios un interés para elegir una carrera que esté vinculada. Estamos más que satisfechos, más que contentos con la información que nos has dado. Estamos seguros que en la ciencia va a encontrar algunos receptores que por conocimiento, y pues celebramos mucho también esta ocasión la Universidad de Texas, la Universidad de Austin, pues participe también en estos eventos. Para nosotros es muy motivador y es uno de los propósitos también del consorcio, que participen especialistas de muchas universidades para que quienes estén interesados en estos estudios en otras de las universidades, de los centros de estudios superiores que participen en el consorcio, se puedan sumar también y puedan también intercambiar contigo alguna información, algún interés, algún conocimiento que pueda ser útil para ambos. Pues muchas gracias a todos, agradecemos su presentación y su presencia y los seguimos convocando para que nos acompañen en esta serie formidable, interesantísima, que nos permite conocer más del planeta y más de sus habitantes en estas pláticas que nos están haciendo el favor de compartir cada martes. Gracias a todos y muy buenas tardes.
Hemos becado a más de 1 millón de estudiantes universitarios.
Más de 84,000 alumnos y alumnas han sido beneficiados con becas nutricionales.
Lanzamos una campaña para apoyar a la Universidad Autónoma de Guerrero, afectada por el huracán Otis.
Este año organizamos nuestra tercera gala musical y nuestra cuarta carrera atlética.
Estamos felices porque más del 87% de nuestros becarios son la primera generación de universitarios en su familia.
Cada año se apoyan a 100 estudiantes de la Facultad de Derecho.
Hemos entregado más de 600 becas a estudiantes de bachillerato junto con Fundación BBVA.
Hemos transmitido 195 documentales y 160 conferencias.
Apoyamos a la Unidad de Investigación en Neurodesarrollo de la UNAM.
Todos nuestros informes son públicos.
Se han entregado más de 50 premios para fomentar la investigación científica.
Con el programa Bécalos, el Patronato de la Facultad de Química y Fundación UNAM otorgaron más de 30 becas.
Con la ayuda de Fundación Carlos Slim hemos otorgado becas de apoyo nutricional y económicas.
Con el programa Hídrico Modelo ayudamos a 340,000 habitantes.
Tenemos 2 premios EFI Social por nuestras campañas publicitarias.
Junto a Grupo Val, más de 2,000 universitarios han sido beneficiarios.
Otorgamos más de 400,000 tratamientos.
Durante la pandemia apoyamos con las campañas Dona un kit y apoya a un residente a estudiantes de medicina de la UNAM.
Durante el confinamiento COVID, donamos tablets para estudiantes de escasos recursos.
Hoy son más de 27,000 solidarios impulsando la educación.
Participamos en la conservación de corales en el Caribe mexicano.
Brindamos más de 4,000 becas de movilidad internacional.
Colaboramos con el Semillatón a preservar el maíz en la Sierra Tarahumara.
Apoyamos el proyecto de conservación de perritos llaneros en la Reserva de la Biosfera de Jano, Chihuahua. Participamos en el proyecto de conservación de la guacamaya roja en el Parque Nacional de Palenque.
Desarrollamos el programa Líderes Transformadores.
Participamos con el Consorcio de Universidades por la Ciencia.
Organizamos 11 ediciones del Foro 2020.
Fundación UNAM resguarda el Palacio de la Autonomía, inmueble histórico de la universidad.
A través del CI ofrecemos cursos, diplomados y talleres.
Dotamos de equipo al Observatorio Astronómico Nacional de San Pedro Martes.
Apoyamos a alumnos de escasos recursos.
Asignado a la opción señalada, institución de asignación UNAM.
¡Sí quedé! ¡Sí quedé!
El sueño se cumplió. Ahora formas parte de la comunidad universitaria y puedes acceder a las becas que Fundación UNAM tiene para ti. Conoce nuestros programas en www.funam.mx y en nuestras redes sociales.